friend or foe, tolerate or destroy. Every single second of your life, your immune system is basically making these brutal binary choices.
It really is. And, when we try to picture a defense system that complex, we usually just imagine pure chaos, right?
Red flashing lights, alarms blaring everywhere, total sensory overload.
Exactly. It's so easy to assume your body operates like this panicked war room.
Yeah.
But when you look closely at the actual mechanics, it's remarkably orderly.
Yeah.
It's basically the ultimate decision tree forged over, millions of years of evolutionary trial and error.
And that is exactly what we are exploring today on this deep dive. So we've got an incredibly detailed set of notes, a slide deck, actually, by Dr. Khushbu Singh.
Right.
It's titled Dichotomies in Immunology. And honestly, what makes this material so brilliant is that it takes this massive, intimidating biological network and just, it breaks it down into a strict sequence.
Yeah, a sequence of this versus that comparisons.
Yeah, exactly. So for you listening, instead of drowning in medical jargon, we're going to walk you through these choices sequentially. You'll actually see the underlying logic of your own survival.
It's a great way to look at it.
Yeah.
Because before a war can even begin, your system has to actually recognize the enemy.
Right. So let's start there. How does the body identify a threat? Because the slides start with this golden rule.
Yes. The rule is, and this is fundamental, all immunogens are antigens, but not all antigens are immunogens.
Which sounds like a riddle at first.
It does, yeah. But to make sense of it, you really have to look at the energy economics of your body.
Okay, break that down for us.
So an antigen is simply any substance your immune system is physically capable of recognizing. It has a shape, right? And a biological sensor can latch onto it.
Right.
But an immunogen, that is a substance that actually provokes your body into launching a full-scale attack.
So reading through the notes, I kept thinking of this like a spark versus a fire.
Oh, that's a perfect analogy.
Like, let's say a tiny, simple molecule enters your bloodstream. The slides call it a hapten.
Right, a hapten.
Your body sees it, the sensors recognize it, so boom, it's an antigen. But it's just a spark without any fuel.
Exactly. That doesn't trigger the alarms.
Because your body doesn't want to launch this massive, exhausting biological war over every single tiny piece of harmless dust you inhale.
Right. I mean, and the energy expenditure of doing that would literally kill you faster than any pathogen ever could.
Oh, wow.
Yeah, so the system waits. It waits for the right chemical signature, the right size, and, the right complexity.
So how does that spark actually start a fire?
Well, if that tiny hapten physically binds to a much larger carrier protein.
Oh, so it suddenly has weight to it.
Exactly. It gains complexity, it crosses that biological threshold, becomes an immunogen, and the fire starts.
Okay, so the fire's started. The threat is officially recognized as an immunogen. But not all threats are created equal.
Not at all.
How does the body decide what level of response to actually deploy?
It looks strictly at the structure of the invader. And this leads to our next major dichotomy from the slides. Thymus-dependent antigens versus thymus-independent antigens.
Or TD versus TI.
Right. And you can think of this as the difference between, say, standard operations and a massive system upgrade.
Okay, I like that. So what's the upgraded version?
That would be the thymus-dependent antigens. These are highly complex, easily degradable proteins. Because they're so complex, the frontline cells, they just can't handle them alone.
They need backup.
Exactly. They have to call in specialized T cells for help.
And from what I understand from the deck, calling in those T cells unlocks some serious biological weaponry. You don't just get a basic attack.
Oh, absolutely not. You get an upgraded custom response. You get memory cells, which is huge.
Meaning your body remembers this exact invader decades later.
Precisely. And you also get something called affinity maturation.
Which sounds intense. What is that?
It's amazing, really. The antibodies your body produces, they actually mutate and tweak their own physical shape in real time to bind tighter and tighter to the target.
Wait, they literally get better at their job while the fight is happening?
They do. They sharpen themselves during the battle. It's incredible. And these are the highly effective IgG antibodies.
That is mind-blowing. But, okay, what about the thymus-independent antigens, the ones that just, don't get that T-cell upgrade?
Those are usually simple, highly repetitive structures. Think of like the sugary capsule surrounding a basic bacterium.
Right.
They don't need T-cell processing. They just slam directly into your B cells, and trigger this immediate blunt force response.
So no specialized sniper rifles here.
No, just basic heavy-duty antibodies called IgM. You don't get memory. You don't get the evolving, sharpening antibodies.
Wow.
It's really just a quick, dirty stopgap measure to hold the line.
So it's a fast response, but it completely fails to help you if that exact same bug comes back next year.
Exactly.
Okay, I have to stop here because the next comparison on threat identification is just, it's wild.
Oh, the superantigens.
Yes. We've talked about conventional antigens, which seem highly targeted, right? But then there are superantigens. And the mechanics of how these work, I mean, it sounds like something out of a sci-fi movie.
It really does. And it comes down to the geometry. The geometry of a superantigen is what makes it so lethal.
How so?
Well, normally, a conventional antigen is carefully eaten by an immune cell, right? It's chopped into tiny little pieces.
Like processing evidence.
Right. And then one highly specific piece is presented perfectly inside a specialized chemical groove.
To the T cells.
Exactly. And it activates maybe 0.01% of your T cells. It's a sniper rifle targeting one specific threat.
Okay, but the superantigen.
A superantigen completely bypasses all that internal processing. It physically wedges itself onto the outside of the receptor complex.
Outside the groove, it forces the cells to lock together whether they actually match or not.
Yes. It forcibly bridges them.
The numbers in the slide for this are terrifying. It says it triggers a massive, non-specific response of 5 to 25% of all your T cells at the exact same time.
Yeah, it's a massive systemic shot.
Oh, wait, let me push back on this for a second. If superantigens trigger up to a quarter of your entire immune army to fire their weapons all at once,
Right.
isn't that complete chaos? Like, doesn't that massive overreaction harm the host way more than it actually harms the pathogen?
Oh, it's incredibly devastating to the host. In fact, that exact massive systemic shock is what causes things like toxic shock syndrome.
Oh, wow.
Yeah. But you have to view this from the perspective of the invading bacteria, that chaos. That is the entire point.
Wait, really?
Yes. By triggering 20% of your immune system to fire blindly in every possible direction, the pathogen is essentially throwing a physiological flashbang into your command center.
A biological flashbang.
Exactly. Your immune system is just so overwhelmed by the sheer volume of screaming alarms that it cannot mount a coordinated specific attack against the actual bacteria.
So they weaponize our own defense system's volume knob against us. That is incredibly sinister.
Evolution is ruthless.
Wow. Okay. So assuming we aren't dealing with a flashbang and a cell has successfully recognized a standard complex threat,
Right.
how does it actually communicate that finding to the rest of the body? Like, how do the troops actually know where to go?
This moves us seamlessly into the domain of the major histocompatibility complex, or MHC for short.
Thank goodness for abbreviations.
Seriously.
Yeah.
Think of MHC molecules as cellular billboards. And the crucial dichotomy here in the slides is between MHC class 1 and MHC class 2.
I love how visual this is. Let's look at MHC class A first.
Yeah.
The notes say it's found on almost every single nucleated cell in your entire body.
Yeah, basically everything.
From a skin cell to a liver cell to a heart muscle cell, it seems like it acts as this constant status report.
That's a great way to think of it.
Right. Like if a normal everyday cell gets hijacked by a virus, it uses its MHC class I billboard to hold up a flag saying, hey, I have been compromised from the inside. Do not try to save me. Destroy me.
That is a perfect translation of the mechanism. It's presenting that internal threat directly to cytotoxic T cells.
The assassins of the immune system.
Exactly, the assassins. But MHC class 2 is a much more restricted distribution.
Right, you won't find it on just some random liver cell.
No, it is only found on professional security forces. Cells like macrophages or specialized B cells, these are your antigen-presenting cells, or APCs.
So if class 1 is a destroy me status report, class 2 is like a wanted poster held up by a roaming security guard.
I love that. Yes.
Like a macrophage is out on patrol, right? It finds a rogue bacteria floating around outside, eats it, and then uses MHC class 2 to say, hey, look what I found in the neighborhood. Let's go hunt down the rest of them.
Exactly. And that roaming security guard presents the wanted poster to helper T cells.
Generals.
Right, the generals who coordinate the broader war effort. But, you know, the really elegant part of this whole system is how the cell physically knows which billboard to actually use.
Oh, interesting. How does it know?
The origin of the threat dictates the internal plumbing the cell uses to process it.
Wait, let me make sure I'm getting this. So the inside of the cell literally routes the trash differently depending on where it came from.
Exactly. It's a highly segregated system.
Right.
If it's an endogenous threat, meaning a virus is already inside your cell using your own machinery to manufacture viral proteins.
Right.
Those proteins are chopped up in what is called the cytosolic pathway. They're processed right there in the main fluid of the cell and automatically routed to the MHC class I billboard.
Ah, so that's the destroy me signal.
Precisely, because the threat came from within.
But if it's an exogenous threat, something the cell ate from the outside.
Then it gets routed through the endocytic pathway. It is trapped inside an acidic little bubble, broken down, and specifically loaded onto the MHC class II billboard.
The wanted poster.
Exactly. The symmetry is just brilliant.
Yeah.
Inside threat equals inside processing equals class 1. Outside threat equals outside bubble processing equals class 2.
The routing system is flawless. Okay, so the billboards are up. The wanted posters are printed and distributed. Who actually answers the call?
The troops?
Yes. This brings us to the cellular army itself, primarily the B cells and the T cells. Now, they both fight, obviously, but their jurisdictions seem completely different.
They view the battlefield in fundamentally different ways.
Huh.
So B cells, which, by the way, develop in the bone marrow, they operate mostly in the fluids of the body.
Okay.
They use a receptor that is essentially just a membrane-bound antibody. This allows them to recognize free-floating, raw, unprocessed threats.
Oh, so a B cell can simply bump into an intact virus floating in your bloodstream, recognize its shape, and just immediately start secreting antibodies to neutralize it.
Exactly. No middleman required. But T cells are much higher maintenance.
Right, they can't just bump into a virus and do something about it.
No, T cells are practically blind to raw pathogens. A T cell, which develops in the thymus, hence the T cell, it absolutely requires that MHC billboard.
It needs the paperwork.
Exactly. It can only see a threat if another cell has carefully processed it, chopped it up, and is physically handing it to the T cell on a silver platter.
Wow.
And instead of secreting antibodies like the B cells do, T cells secrete cytokines.
Which are basically chemical messengers that direct other cells on how to fight, right?
Exactly. They are the commanders.
Although, looking at the notes here, there is a fascinating exception to this rule. It mentions a subgroup called gamma delta T cells.
Yes.
Yeah, outliers.
Yeah. If standard T cells are these strict rule followers who demand the proper MHC paperwork, these gamma delta cells seem to completely ignore the chain of command.
They really do. They don't require the MHC billboard at all. And they recognize completely different types of non-protein molecules.
So what exactly are they doing? They're essentially the evolutionary bridge between your blunt, innate immune system and your highly targeted adaptive immune system. They patrol the borders like your gut and your skin.
Like a rapid response unit.
Exactly. They're a black ops unit that doesn't need to wait for official orders to start taking out targets.
Black ops in the gut. I absolutely love it. Okay, so now, where do these troops actually train before they deploy? We've got primary and secondary lymphoid organs.
Right.
And the tagline of how these organs develop is surprisingly strict in Dr. Singh's slides.
It is. The primary organs are your boot camps.
Mm-hmm.
In humans, that's the bone marrow for B cells and the thymus for T cells.
And they develop really early, right?
Very early,
Mm-hmm.
while you are still an embryo. But the most crucial feature of a primary organ is that it is a completely safe space.
Safe space? How?
It is entirely shielded from actual real-world antigens. It's a highly controlled environment where young immune cells are tested to ensure they won't accidentally attack your own body.
Oh, that makes sense. You don't want friendly fire. And then, bizarrely, the thymus boot camp actually shrinks and loses function after puberty. It just closes up shop.
Because by the time you reach adulthood, you've already built your standing army of T cells.
So it's just efficiency?
Exactly. The body no longer needs to expend the massive energy required to run this huge training facility. Conversely, the secondary lymphoid organs.
Like your spleen and the lymph nodes in your neck?
Right. Those persist throughout your entire life.
Ugh.
Those are the actual reactive battlefields where the mature troops deploy and wait to encounter the enemy.
And there's this wild clinical example in the deck that proves who does what. It says, if you were to surgically remove the thymus boot camp from a newborn baby.
Right, a neonatal thymectomy.
Their circulating immune cells would practically vanish. Their body would completely lose the ability to reject a foreign tissue graft, like a skin transplant.
Because you've eliminated the T-cell training facility, the generals and the assassins are gone. But notice that the baby's basic antibody levels wouldn't drop that much. Now, if you look at birds,
Birds.
yes, birds have a special organ called the bursa of Fabricius, which is their primary boot camp for B cells.
Okay.
If you surgically remove the bursa from a newly hatched chick, tissue graft rejection remains totally normal because the T cells are fine. But their antibody production falls off a cliff.
It's a perfect double dissociation. It proves exactly which boot camp is responsible for which weapon system.
Exactly.
So let's talk about those weapons. Our troops are deployed in the lymph nodes. They've spotted the enemy. What physical tools do they use to destroy a pathogen?
Well, let's look at the complement system.
Okay.
The complement system is an incredibly lethal cascade of proteins just floating in your blood, waiting to be triggered. And we see a clear dichotomy in how they are triggered. The classical pathway versus the alternative and lectin pathways.
Right, and the crucial difference here seems to be timing and targeting.
Yep.
The classical pathway is like a highly targeted missile strike. But it's slow, right? It requires a specific antibody to bind to the specific pathogen first.
Exactly. And because it takes your body time to manufacture those custom antibodies, this pathway doesn't fully activate until 7 to 10 days after you get infected.
Which feels like a lifetime during an infection.
It is. But the alternative pathway, however, doesn't wait for custom antibodies.
It's immediate.
It acts like an ever-present microscopic tripwire. It simply recognizes the generalized weird textures found on the surface of most bacteria and instantly triggers the cascade.
So it's an immediate response?
Yes. But regardless of which pathway pulls the trigger, the end result is the same. These proteins assemble themselves into a microscopic cylinder and literally punch a physical hole through the bacterial membrane, causing it to explode.
Just blowing holes right in the hull of the ship. Incredible.
It's brutal.
Now, coordinating all of this requires massive communication. The notes contrast hormones and cytokines. They're both chemical messengers, but they differ wildly in functional redundancy.
Yes, this is a very important distinction.
Hormones, like insulin or thyroid hormone, have very specific sources, specific targets, and zero functional redundancy. If your pancreas stops making insulin, there isn't another hormone that can step in and do the exact same job. You are in serious trouble.
But cytokines are the exact opposite. They have massive functional redundancy. Multiple different types of immune cells can produce the exact same cytokine, and multiple different cytokines can trigger the exact same response in a target cell.
Why build a system with so much overlap, though? Like, isn't that inefficient?
In peacetime, maybe. But your immune system is built for war. And in a war zone, communications get jammed. Viruses are incredibly tricky. Many of them have evolved ways to block specific immune signals.
Okay, I see where you're going with this.
Right. If a virus blocks cytokine A, your immune system has cytokines B, C, and D ready to step in and deliver the exact same attack order. The redundancy is a built-in fail-safe survival mechanism.
That makes perfect sense. Now, speaking of the brutal realities of war, let's talk about how cells actually die on the battlefield, because there's a huge difference between apoptosis and necrosis.
Absolutely.
If you want to understand how elegant your body can be, look at apoptosis. It's an active, intentional process. A cell realizes it's infected or terminally damaged, and it initiates a self-destruct sequence.
That's highly regulated.
It's like taking down a condemned building with a quiet, highly controlled demolition. The cell shrinks, the DNA is neatly packed up into little manageable bubbles, and surrounding cells quietly eat the debris, and crucially, no alarms are triggered.
Exactly. Necrosis, on the other hand, is a messy, unannounced wrecking ball.
Just chaos.
Complete chaos. It is a passive process caused by severe trauma or toxins. The cell swells up, the membrane ruptures, and all of its highly acidic, toxic internal contents just spill out into the surrounding healthy tissue.
And the body hates messy.
Because that spilled debris is entirely unnatural. It is exactly the kind of biological trash that triggers the microscopic tripwires we just talked about. Necrosis triggers massive painful inflammation. Apoptosis prevents it.
Wow. Okay, before we leave the arsenal, there is one structural oddity we have to mention.
Oh, the antibodies.
Yes. When we look at the antibodies, the IgGs, most of them are built pretty similarly. They have a standard hinge region that gives them a bit of flexibility, like a normal neck.
Right.
But then there's IgG3.
Yes, IgG3 is built very differently. While a standard antibody hinge might have, say, 15 amino acids, IgG3 has a massively elongated, flexible neck made of 45 amino acids.
45. It's like a giraffe compared to a horse. Why does it need a neck that long?
That extreme flexibility allows it to reach deep into awkward hidden crevices on a pathogen that other stiffer antibodies simply cannot access.
Oh, wow.
It is an incredibly effective specialized weapon. But, that long neck also makes it physically fragile. It's prone to breaking down quickly in the bloodstream.
So it's a highly flexible tank, but it requires constant maintenance.
Exactly.
Okay, we've traced the entire process, the recognition, the routing, the army, and the weapons. But, and this is the dark side, what happens when this exquisitely balanced targeting system makes a mistake? When the dichotomies misfire?
That brings us to hypersensitivity and autoimmunity, the cost of friendly fire.
Yeah.
We classify hypersensitivity into 4 types, and they perfectly mirror the mechanisms we've discussed today.
Right, like type I is your classic immediate allergic reaction driven by an antibody called IgE. It happens in minutes.
Right. But then there's type I, which doesn't use antibodies at all. It is entirely run by those T cells taking their sweet time to coordinate an attack.
Which is why it's delayed.
Exactly. If you ever brushed up against poison ivy and wondered why the itchy red rash didn't actually show up until 3 days later,
Oh, yes.
this is exactly why. Your key cells were slowly mobilizing for a completely unnecessary war against a plant oil.
Unbelievable. But the most profound contrast in the slides, I think, is between type 2 and type 3 hypersensitivities.
Yes, mechanically they are fascinating.
Huh.
In a type 2 reaction, your antibodies mistakenly target an antigen that is physically attached to a specific tissue in your body, like, say, your red blood cells.
Okay.
The immune system acts like a sniper, taking out that specific cell type, so the damage is highly localized.
But type 3 is a completely different beast.
Oh, entirely.
Yeah.
In type 3, your antibodies attack soluble antigen stuff just floating freely in your blood.
Right.
When the antibody grabs the antigen, they form this heavy, microscopic clump, an immune complex, and these clumps just float through your circulation until they get physically stuck.
Stuck where?
Usually in tight filtration spaces,
Yeah.
like the delicate tubes of your kidneys or
Right.
the lining of your joints. And once they get stuck, they trigger those complement tripwires we mentioned and cause massive inflammation right there.
Wow.
The damage isn't localized to the source of the antigen. It is collateral damage caused by the floating debris. It is essentially carpet bombing.
So it destroys the joint not because the joint is the target, but because the garbage got stuck in the joint.
Exactly.
That is tragic. And this leads to an incredibly deep realization about autoimmune diseases.
Yes.
The notes divide autoimmunity into organ-specific, where the attack is localized to one place, like the thyroid, and non-organ-specific, where the attack is systemic, like in lupus.
Right.
But there's a detail here that genuinely stopped me in my tracks.
The cancer link.
Yes. It says that organ-specific autoimmunity drastically increases your risk of developing cancer specifically in that organ.
Yes.
Is the body literally exhausting its own tissues into mutating through constant friendly fire?
That is exactly what is happening. It is a profound and, frankly, dark pathological mechanism.
Walk me through that.
Okay, imagine an organ is under constant localized immune attack. The cells are continuously being blown apart by the complement system or cytotoxic T cells.
Right.
To survive, that tissue must rapidly and continuously divide to replace the dead cells. But every single time a cell divides, it has to copy its DNA. And there is always a microscopic risk of a copying error.
A typo. So decades of forced rapid cell turnover just mathematically increases the probability of a deadly typo.
Precisely. Chronic inflammation drastically increases the statistical probability of a mutation that leads to cancer.
Wow.
And in non-organ-specific diseases where the entire immune system is constantly activated in a systemic war,
Right, like lupus.
the immune cells themselves are forced into overdrive. This is why systemic autoimmune diseases carry a higher risk of systemic cancers of the immune network itself, like lymphomas or lymphoreticular neoplasia.
Wow. That is heavy, but it makes total mechanical sense. The friendly fire literally forces the tissue to mutate just to keep up.
It does.
So we've gone from the very first spark of recognition, through the elegant sorting pathways, to the deployment of the troops, the firing of the weapons, and finally the tragic cost when the targeting system fails.
It's a journey.
It really takes the chaos of biology and reveals this stunning logical framework.
It's a remarkable system of checks, balances, and, as we've seen, profound dichotomies. But, I want to leave you with one final thought to mull over.
Okay, let's hear it.
Throughout this entire exploration, we've relied on these stark, ancient evolutionary lines, right? T cells versus B cells, inside processing versus outside processing.
Right, the rules of the game.
But consider how modern medicine is evolving today. Take artificially engineered CAR T cell therapy for cancer, for instance.
Wait, what exactly is a CAR T cell doing that breaks these rules?
Well, we are currently taking a patient's T cells into a lab and genetically engineering them to possess the direct, blunt force antigen recognition of a B cell.
Wait, really?
Yes, but keeping the lethal targeted killing power of a T cell. We are completely bypassing the NHC billboards. We are crossing the streams.
We are taking the B cell's ability to just bump into a target and strapping it onto the T cell's assassin machinery.
Exactly. So the question is, are human interventions beginning to blur the very evolutionary lines and dichotomies we just spent this time learning about?
Wow.
As the future of immunology unfolds, we aren't just studying these biological forks in the road anymore. We are beginning to pave our own artificial shortcuts right through the forest.
We aren't just reading the command center's rulebook, we are actively rewriting the code. That is an incredible thought to leave on. Thank you for joining us on this deep dive into the ultimate biological decision tree. Keep questioning the world inside you, and we'll catch you on the next one.